My name is Maricarmen Hernandez Rodriguez. I'm from Escuela de Medicina, Instituto Politécnico Nacional from Mexico City. I belong to the Cell Culture Laboratory. I'm very grateful for the opportunity to present my research work titled Drug Repurposing of Histamine and Methyltransferase Inhibitors and Their Evaluation in a Scopolamine Induced Amnesia Model during the 29th Symposium of Bioinformatics and Computer-Aided Drug Discovery. Well, histamine is a neurotransmitter which exerts a wide variety of functions. It is well known that histamine regulates the allergic responses, regulates the gastric acid liberation by the parietal cells in the stomach. However, we will focus on the effects produced by histamine in the central nervous system. In the brain, histamine is involved in many essential physiological processes, such as the regulation of the sleep-wake cycle, learning, memory, stress responses, etc. In addition, recently it was demonstrated that histamine allowed the degradation of proteins involved in the development of Alzheimer's disease, including amyloid beta peptide and hyperphosphorylated tau protein. For this reason, it's important to note that indeed lower activity of the histaminergic system has been associated with neurological disorders, including Alzheimer's disease, narcolepsy, Tourette syndrome, depression. For this reason, several research groups have been focused on increasing histaminergic system, which appears as a promising approach to treat the aforementioned diseases. Histamine is produced by the histaminergic neurons located in the tuberomamallaris nucleus in the hypothalamus. Histamine is produced by the action of the histidine descarboxylase, which employs histidine as a substrate. After histamine is released to the synaptic cleft in the extracellular space, it can interact with several receptors, including histamine 1 receptor, histamine 2 receptor, histamine 3 receptor, and histamine 4 receptor to exert their physiological functions. However, in the brain, the only pathway for the inactivation of histamine is produced by the histamine and methyltransferase, or HNMT, enzyme, which inactivates histamine by converting this to telemethylhistamine. In comparison to peripheral tissues where histamine is inactivated by deaminoxidase. The increasing histamine levels in the brain can be achieved by HNMT inhibition, as it was demonstrated by metoprene. Metoprene is a potent HNMT inhibitor, which has been shown at inhibitory concentration 50 of 100 nanomolar. Indeed, it was demonstrated that the administration to Wistar rats to metoprene at a dose of 10 milligrams per kilogram results in the increase of histamine levels in the brain, being the highest at about 5 hours of administration, which correlates with the drug peak of metoprene. Indeed, most of the biological effects of increasing histamine in the brain have been demonstrated by employing metoprene, including antinociception, suppression of energy intake, improvement of cognitive function. Interestingly, the effect of metoprene and the consequently increased levels of histamine in the brain showed an improvement in memory performance, which has been demonstrated by administration of metoprene in a mouse model of amnesia induced by scopolamine. And interestingly, these effects were reverted by the administration of an antagonist of H1 receptor. However, only a small number of molecules have been identified as HNMT inhibitors. Furthermore, only a few of them have been studied due to their poor penetration into the blood-brain barrier and due to their toxic effects. For this reason, the design and the development of novel HNMT inhibitors appears as a promising approach. For this reason, our research group focused on perform a drug repurposing of HNMT inhibitors. To achieve this goal, firstly, we perform a series of docking studies employing Autodoc 4.2 to perform docking studies of a database of 185 compounds, which includes FDA approved drugs for the treatment of neurological diseases. These compounds were employed to perform docking studies with the three-dimensional structure of HNMT. It's important to mention that only drugs employed for the treatment of neurological diseases were included in the study because to ensure that the compounds can cross the blood-brain barrier. After that, the compounds with the highest affinity to HNMT were selected to perform molecular dynamic simulations of the protein ligand complexes to evaluate the stability by the free energy calculated and the non-bonding interactions. After that was performed an HNMT inhibition assay by in vitro studies to corroborate the effects on the HNMT activity. After that, the compounds were administered to Western rats to extract their brains after 3 hours of intraperitoneal administration to determine the changes in the histamine levels. And finally, the effects in memory for both compounds were studied by the novel object recognition test. This test is based in the spontaneous tendency of rodents to explore more time or to spend more time exploring a novel object in comparison with a familiar object. And this behavioral is used to evaluate recognition memory. According to the obtained results, it's possible to observe that taking into account the values of the Gibbs free energy of the interaction with the HNMT enzyme, it was possible to select two compounds, dihydroergotamine and vilazodone, which showed the high high affinity to the HNMT enzyme or the lowest Gibbs free energy. It's important to mention that dihydroergotamine is employed to treat migraine, while vilazodone is employed to treat depression. In figure 1, we can see the chemical structures of the selected compounds. After that, the protein ligand complexes of both selected compounds were submitted to 50 nanoseconds of molecular dynamic simulations to evaluate the free energy binding and the non-bonding interactions. As it can be seen in table 2, vilazodone show a lower free energy in comparison to dihydroergotamine, showing more stable complexes. This fact can be explained by analyzing the non-bonding interactions established by the compounds with the histamine binding site of HNMT. As it can be seen in figure 2, the interactions of the non-bonding interactions of the selected compounds with the histamine binding site of HNMT. It has been demonstrated that compounds which act as HNMT inhibitors interacts with interacts by establishing three important hydrogen bonds with specific amino acid residues in the histamine binding site of HNMT, which are represented by glutamate 28, asparagine 283, glutamine 143, by establishing hydrogen bonds. In addition, it was reported that along the cleft of the histamine binding site, there are the lateral chains of amino acid residues, aromatics, which allows the establishment of PP interactions with the inhibitors, which increase their affinity. As it can be seen in figure 2, pilazodone established the hydrogen bonds with the aforementioned residues in addition to PP interactions with the aromatic lateral chains of the amino acid residues along the cleft of the histamine binding site in comparison with dihydroergotamine. This can be due pilazodone showed a more flexible structure in comparison with dihydroergotamine. After that, we evaluate the effect of both compounds in an in vitro site of inhibition of HNMT, where it's possible to observe that both compounds inhibit HNMT at a micromolar range. However, the inhibition was bigger for pilazodone in comparison with dihydroergotamine. After that, we perform the administration of both compounds to Wistar rats by intraperitoneal route, dihydroergotamine at a dose of 1 milligram per kilogram, and pilazodone at a dose of 4 milligram per kilogram. And after 3 hours, we extract the brains to quantify the brain histamine levels. As it can be seen in figure 3, pilazodone and dihydroergotamine increased histamine levels. However, the effect was more pronounced for pilazodone in comparison to dihydroergotamine. Finally, the effects in memory were evaluated in a novel object recognition behavioral assay. In this assay, we employ control Wistar rats as a control. We remember the exposition to a familiar object. However, when we expose the rats after a period of time, we can remember the exposition to the familiar object and spend more time evaluating or exploring the novel object. When we administrate the scopolamine, this compound impairs the memory, and we can evaluate in this model the restoration of the memory with the administration of compounds. In this case, as it can be seen in figure 4, the administration of pilazodone and dihydroergotamine allows to restore the memory and remember the exposition to the familiar object, similar to the control rats, which is evidenced by the discrimination index. In conclusion, in this work, drug repurposing of HNMT was achieved by employing computational studies. In addition, HNMT inhibitory effect of pilazodone and dihydroergotamine by in vitro assays were demonstrated. In addition, pilazodone and dihydroergotamine increases histamine brain levels and improve memory in the novel object recognition paradigm in the scopolamine induced amnesia model. It's important to mention that at this stage of their research, we are evaluating the reversibility of the effect by blocking the H1 receptor. Thus, we allow to demonstrate that the improvement in memory is mediated by the increasing of the histamine levels exhibited by dihydroergotamine and pilazodone. Finally, I want to say thanks to our research team by PhD Elvia Mera Jimenez from Mexico, PhD Teresa Solek from Poland, which support us with the realization of molecular dynamics studies. PhD Marinés Nicolás Vázquez, PhD René Miranda Rubalcaba, and of course our students, Paola Gabriela Hernandez Pérez and Luis Gerardo Oliva Ramírez. And in addition, this work was supported by Consejo Nacional de Humanidades, Ciencias y Tecnologías from Mexico with a grant number 320862 and Secretaría de Investigación y Posgrado with 3 grants. Thank you for your attention.
Okay, thank you very much, Maricarmen, for your nice presentation. Thanks a lot. We have still 3 minutes, but we do not have any questions.
No, there is one question in the chat.
But yeah, I would like to ask a couple of questions also. If or you want, Vladimir?
No, there is one question in the chat.
In the chat, yes.
Yes.
I see now there is. What is the difference between carbon-hydrogen bonds and conventional hydrogen bonds in 2D interaction diagram, slide 7?
Okay, carbon, hydrogen bonds and conventional. Well, it's similar. Dr. Teresa only wants to highlight the atoms which forms the atoms we are involved in the formation of hydrogen bonds. Is the only difference. He wants to highlight the type of atoms which enables the hydrogen bonds. It's all.
Mm-hmm. Okay, thank you very much. And I would like also to say you because one of the reasons of your essay was to find some new inhibitors because the previous one were very toxical.
Yes.
Yeah, and what about these inhibitors which you found now after docking and molecular dynamics? Can you say anything about this property?
Yes. We, of course, as you mentioned, there are a few Hnmt inhibitors, although this enzyme was described a long time ago. And nowadays, there are some inhibitors, and as you mentioned, most of them exhibit toxicity, high toxicity. For example, metoprene has high toxicity to the blood cells, and these characteristics avoids that can be used in for example, in patients. In addition, other compounds cannot cross the blood-brain barrier. This is the first intention to search novel Hnmt inhibitors. We perform drug repurposing employing only FDA-approved drugs which are in the market employed for the treatment and other diseases in this case. And in addition, we only employ drugs employed to treat neurological. In this case, we are secured that across the blood-brain barrier and that are safety because they are drugs that are approved by FDA.
Okay. Thank you very much.